Thank you for the introduction, Felicia. Yeah. Emeritus Professor means retired. Well, the topic I'm going to talk about is actually the emerging field of cell-based therapy, immunotherapy. At this stage, this is coming, right? I hope that at the end of this talk, some of you may be interested to develop this field, or if you decide to have a mid-career change, this might be a promising area to go into. So first, as usual, I have to declare myself. I'm the medical advisor to Cytoprotics, which is actually a clinical research organization established for cell-based therapy. And also a lab director to Crown, which is a stem cell banking with the largest GMP facility in Southeast Asia. Now, the reason why I have to declare because both are startups, which I was involved for the last 20 years. So it's not something the last few years, it's actually over 2 decades. Okay, next please. Now, what is immunotherapy? One of the definitions is to use an agent to suppress or enhance the immune system to fight cancer, not just cancer, infection and other diseases. Example, you probably heard of just now, the immune checkpoint inhibitors. There are lots of monoclonals in the market, immune modulators, vaccines, and cells. Cells, the last one. Cell-based immunotherapy, essentially to use our own immune cell, such as T cell, NK cell, macrophages, dendritic cells, and tumor-infiltrating lymphocytes. Many more, but these are the ones already being established. Now, this is not new actually. Long time ago, if you look at the literature, you probably, even the textbook will tell you about Dr. Cooley's toxins. He actually observed patient who has got this skin infection. Cancer patient with skin infection, some of them actually, after the skin infection, the cancer actually regressed. Right? In fact, he actually went ahead to do experiment and infect them with these toxins. That's why the name is named after him, Coley's toxins. But this, with some success, of course, but many of them don't. So this has been long forgotten, essentially because the successes due to surgery, radiotherapy, and chemotherapy. Now, I'm a hematologist actually, my profession. So Dr. Thomas is actually, we consider him as the father of bone marrow transplant. He's actually the pioneer in cell-based therapy. Now, this method, when he introduced bone marrow transplant long time ago, when he actually applied for grants, he couldn't get all the grants. He's actually in America. And then everybody is telling me doing a very dangerous experiment. But he persisted because his animal studies show very good success using dogs to do bone marrow transplant. Now we know that actually bone marrow transplant is actually a form of immunotherapy through hematopoietic stem cell transplant, which then developed into the whole immune system. And the new immune system that has been engrafted in the human body will then guard against the emergence of cancer. That's the basis of it. Yeah. So now, in fact, more than 1 million patients have now received this type of treatment, and half of them actually are cured of the disease. So this type of treatment is not just looking for response, looking at all this Kaplan-Meier plot, but it's actually curing them. So in fact, for his work, he was conferred a Nobel Prize. Yeah. However, bone marrow transplant is a very, very toxic procedure. It carries a mortality of 10% in the best hand. What it means is that 1 in 10 is going to die because of the procedure. Right? And there are other side effects, serious side effects. For example, the graft, which is the stem cell that is transplanted, can actually attack the donor, right? And then produce a lot of unpleasant side effects. Next, please. So then, because of that concept, if you use hematopoietic stem cell, we transplanted and therefore established a whole new immune system in the recipient. Why can't then we use specific cells to treat specific cancers? So that actually, all these new cells, which are actually component target cells developed for the whole immune system, right? Which I often refer to as the Ministry of Defense within our body, that actually guard us against all the pathogens that is floating around. Yeah. So in fact now, FDA, many of the researchers have regarded all these immune cells, if you want to do it, you want to use it as a tool for treatment, and it has to undergo the same rigorous validation, experimentation like any drug. So that's why it's now referred as a live drugs that's going to be used. So this picture just summarizes what are the cells I'm going to go to, right? So T cell, NK cell, macrophages, and dendritic cell. Okay, I will go one by one just to give you the background. Next one, please. Now, first the T cell. T cell based immunotherapy. So T cell, we all know that it's actually, they will recognize specific antigens of the tumor. Okay, and then the interaction will then result in tumor kill. I won't go into the detail. Now, among the T cells, which have been well studied, the tumor infiltrating lymphocyte, TIL. Now, what happened is that people observe over many years already, how come tumor has got lymphocytes in it? What are they doing there? So what they have found is actually, these are all lymphocytes trying to do their job, but obviously not successfully. So then the question is, if they are not successful, then can you sort of isolate them, expand them, and give it back to the patient? Since they are able to infiltrate the tumor, right? Perhaps they can, if you expand them in number, perhaps they can do the job. So that's the idea behind it. Then of course, you can actually modify, because the T cell, right, recognize specific antigen through their receptors. So you can actually engineer the receptor. One of the engineering fit is called the chimeric antigen receptors, so-called CAR T cell. You probably heard about it in the newspaper. Right? And the other one, of course, a T cell that has actually got T cell receptor. So these are the three groups of cells actually being developed, okay? And through their unique mechanism are able to tackle the tumor cells. Next one. All right, so this picture actually shows you the process of producing the tumor infiltrating lymphocyte. Right? I won't go through the detail. So essentially, you just take it from a patient, you expand it outside, right? And then after that, you give it back to the patient after conditioning. And in fact, I was quite surprised the other day. I just read from the news, the first FDA-approved products for melanoma. So melanoma is a very popular cancer. I'm sure the immune checkpoints was also one of those, Jimmy Carter's, isn't it? So one of the success using immune checkpoint. Then the same thing, melanoma also is, able to use the TIL that is infiltrated the melanoma, you isolate them, you expand them and give it back to them, you can do the trick. Yeah. So in fact, because of that, FDA actually, this one is US FDA. FDA approved it just last month. Yeah, last month. Now it's June. Next. Now the next cells is CAR T cells. So CAR T cell is actually, you engineer the T cell with an artificial receptor called CAR, right? And they can recognize specific antigen, such as what has been already been commercialized, CD19, CD20, for example. These are all B antigen. So the CAR receptor can recognize this specific antigen. Okay? And in fact, over the last 10 years or plus, there are many generations of CARs that improve on it so that it can have better efficacy, less toxicity, right? So that's the, and then most important is that they must stay around in the body after injection, all right? So increased efficacy and so in order to engineer it so that they have increased efficacy and fewer side effects. So that this cartoon just illustrates the process of taking patient cells, modify it, and give it back to the patient. Next slide. Now, these T cell receptors, similarly, you can see the same thing, but it's a different receptor. Okay, but the only problem with this T cell receptor is MHC dependent. That means that you have to have the histocompatibility complex to be there, right, to work. But certainly that will be a disadvantage. However, by doing it, somehow they have fewer side effects. Next one. So now, what happened to this T cell? Now, just we have gone through the clinical trials website to search for that information, and then in fact, there are many trials already being done. Okay. Now, the only problem is that these cells are very good for blood cancers, but they are not good for solid cancers. Okay. Yeah. So this just. Next one, please. Now, these are the key milestones for a particular one, the CAR-T, because this is the one that is in the news. And that's the most expensive immune cell on earth. So, in fact, it was done a long time ago, already been conducted, right, experimentally. And of course, the one that came out in the news is the first pediatric cancer patient with leukemia, Emily Whitehead, came out in the news. And you know how much it costs to receive that cost? It is $500,000 US, just one cost, but cure her. Right? So in fact, this is just a picture. So more than 30,000 patients have now received CAR-T worldwide, okay, including Malaysia. And in fact, the laboratories in Cyberjaya actually produce CAR-Ts as well. Next one. Okay, that's her picture. That is, she received it in 2012. This picture just recently been released. Next slide. Now, what are the limitations? The limitation is that patients still relapse, some of them, right? And lack of persistence, and the things that make concern to the patient is that the emergence of new clones, a T cell cancer. So in other words, you get treated and you're cured, but there's a potential there that there's another new cancer. Right? So much so that now FDA have actually, make all the companies to say you must put a label there to inform the patient, saying that although you've got a high chance of getting cured with this treatment, but there's a small chance that you may have a second cancer. And then of course there are life-threatening side effect, but which to us is no problem because they are all manageable. Next. Now the next group of cells is the NK cells. I try to hard it up. NK cells, right? NK cells are not the adoptive immune cell, but it's an innate immune cells, right? So they actually all designed by nature to destroy, transform targets, so-called non-cell. They destroy the non-cell of the human body. So they are actually important, more important than T cell in the early tumor elimination. That means that if there is a mutant cells occurring in our body, the first one is going to be tackled by NK cells. And the good thing about what studies have shown is that they are safe and they do not have those severe side effects of T cells. So, and it can be engineered the same like CAR-T to CAR-NK, right? So that they are more specific in their killing. Okay, next one. So this is just a cartoon to summarize which are the potential sources where you can get NK cells, and then how you manufacturing it and give it back to the patient. Next one. So you can be either autologous or allogeneic, right? And then, So then, of course, they've always got this problem. They are always, whatever cell therapy you do, you always encounter some problem which you need to overcome. Next one. All right, and then like T cell, you can actually introduce CAR in NK as well, so that it will be more targeted. Right? The only problem, of course, for hematology cancer, no problem, because we do know their antigen. But for solid cancer, right, breast, lung, whatever, it's so complex, right? So that's why there will be a problem to design CAR. Now the good thing of what's been found is that this NK cell, all right, they can actually infiltrate the tumor microenvironment and overcome the tumor microenvironment. Because of that, the researcher thought that it might be a very promising tool to develop for solid cancers. Next slide. So what happened? Have study been done? Yes, there are many also. But again, like T cell, blood cancers. Sorry. Not so much of solid cancer. However, clinical trials for solid tumors are now underway. Right? Okay, and in fact, there are 2 human trials for CAR-NK completed in phase 2, and the results are not inferior to what is observed in CAR-T. Right? So in view of their tumor, their ability to infiltrate the tumor microenvironment, so researchers are actually interested to explore, see whether you can combine CAR-NK and CAR-T for a better effect. So what are the limitations? They have certain advantages because it's easier to sort of produce, but in terms of engineering, it may be technically more what they call demanding. And the other problem is like T cell, the persistent after infusion, they live very short. So therefore, you want the cells to remain in the body longer to achieve the what they call tumor killing. And of course, we have got a problem with all this solid cancer. You don't have, it's so complex, right? So more work needs to be done. Now, I just want to bring up this one, cytokines induced killer cell. It was for a while very popular in China, right? So you can actually take a sample of the blood, all right, and then grow these cells, right? And then essentially there are 2 mixtures of cells, the CD3, CD56 cell, and the CD3 cells. And in other words, there are T cell and mixture and K cell, so it's called CIK cell. So it has been shown, sorry, to be active in a variety of tumors, and in fact, about 1 decade ago, it was extremely popular in China. Right? So, but unfortunately, because of poor QC in cell production, over commercialization, and there are a number of patients died because of the infusion. The Chinese government actually put a stop to it in 2016. But now, after they have put all the regulation in place, there seems to be an emerging new interest in this area. Okay, next slide. So this is just a picture to summarize. Essentially, all cell therapy are the same. Take on the patient, it is autologous. Take on the patient, expand, engineer, and give it back to the patient. Now, all work macrophages. So macrophages is also another innate immune cells. Right? There are various subtypes. Major subtype M1, M2. M1 is pro-inflammatory, M2 is anti-inflammatory. Okay? They are heterogeneous, so they are now being explored. Next slide. Now this cartoon actually summarizes what potentially they do for treatment purposes. Next slide. Again, you look at a clinical trial, they are already being carried out, okay? But the only thing is that, again, is not for cancer, solid cancer, okay? And then, largely, what from the clinical trial is that they are being exploited to target disease require regeneration, such as the list of conditions there. Next slide. Dendritic cell, right? Next slide. Yeah. Okay, the last cells I'm going to mention is dendritic cell. All right? Dendritic cell first discovered by Ralph Steinman, and he was also conferred a Nobel Prize for his discovery. It's also another innate immune cell, right? Serving as the most capable antigen-presenting cells to the immune cell. In other words, is like, they are the information collecting cell that present the antigen to the T cell or NK cell. Okay. So they achieve, yeah, okay. So this is actually 20 years ago I played with it. So these are the original true figure and how we generated dendritic cells in the lab. Unfortunately, the outcome for all the clinical study for a dendritic cell have been very disappointing. Next slide. Okay, so again, this picture illustrates how this can be done. Yeah. I won't go into the detail. So this is a form of so-called therapeutic vaccination compared to whatever vaccination, prophylactic vaccination people use for HPV, etc. This is actually using DC to interfere or kill the tumor cells. Next slide. Is there any trial? There are many trials. So all I can say is that the success rate of DC vaccine is extremely low. That's why I also abandoned it and go to some other cells. But there are still people die-hard who believe that they have a role. So studies still continue. Next slide. So the concluding remarks I want to make is that because of all this research and clinical trial, cell-based immunotherapy has emerged as an effective alternative when conventional therapy fails. And investigators are now moving a step forward to see whether this cell therapy can actually incorporate the traditional conventional therapy to get a better results. Right? And GMP-grade facilities are now available. So this cell has no problem being produced, but the only problem is you have to undergo the rigorous study to finally get NPRA to approve for market authorization. So it can be a promising new direction for cancer treatment. I think that's all from me. So thank you very much.